We have to remember that these studies are studies of association, so they do not prove causality. And it's always tempting to think that we have found the culprit, but at the same time, it can be much more complicated. When we started looking at pediatric MS, I realized that it was really a unique opportunity to study environmental risk factors for the disease. I still think that at some point we'll be able to approach a more personalized prognosis in terms of risk to develop the disease.
Hello, I am Emmanuelle Waubant, a neurologist at the University of California, San Francisco. I'm a professor of Neurology and Paediatrics.
And I'm Brett Drummond. You're listening to the ECTRIMS podcast, the official podcast from the European Committee for Treatment and Research in Multiple Sclerosis. MS is a complex and multifaceted neurological disorder with a spectrum of clinical manifestations and disease trajectories. We've known for a long time that while MS definitely has a genetic component, it is not a classic genetic disease. For this reason, a large amount of research has been done to also identify environmental factors that must also be involved. Despite the considerable amount of foundational work that has been performed, new findings are still being made regarding both the genetic and environmental aspects of the disease and how they interact. My guest in this episode, Emmanuelle, is a highly published and renowned researcher in the field of MS risk factors. In this episode, we're going to be delving into the latest findings on the role that environmental and genetic risk factors play in MS. Emmanuelle, thanks for joining me.
My pleasure. Thanks for inviting me.
So I guess to start, we're obviously going to go deeper into both of these aspects, but can you provide a bit of a high-level overview of our current understanding of MS pathogenesis, focusing on how the autoimmune mechanisms sort of interact with genetic predispositions and environmental triggers?
Sure. So what I can do is mostly discuss an overall simplified overview of what we think may be happening, as so far the topic of our discussion today, the interactions between genes and environmental factors in MS, have been understudied, and the interactions are likely quite complex. So on the one side, we have more than 200 genetic variants that contribute to 20 to 30% of MS susceptibility. The main one is called HLA-DRB1 in white, and all the other genetic variants have actually a much smaller contribution. On the other side, there's a few environmental risk factors that have been consistently reported through different studies to increase the risk of MS. And that includes prior infection with HBV, sun exposure or vitamin D status, cigarette smoking. There are probably more environmental risk factors, but the study of these risk factors is only retrospective in MS, basically, or mostly retrospective. And thus it is challenging to identify and quantify these factors years after exposures. And we need to rely on questionnaires, or sometimes when we're lucky, data that have been accumulated prospectively. So several groups have started to study genetic and environmental interactions in MS. But one of the challenges with these type of studies is that they have to be very large in order to not miss very small or moderate interactions, not the very small, but just small or moderate. So the size of the published studies so far is actually moderate when you consider that you would need thousands and thousands of participants in the study to really get a good grip on gene-environmental interactions. The size of most of the published studies is several hundreds. So what happens is that you cannot look at all the risk factors, all the genetic variants, and all the environmental risk factors that have been identified. And you have to select basically the strongest genetic risk factor of interest, plus maybe a few genetic variants that are on the pathway involved with the environmental risk factor. in order to narrow down what your start with and also study only a few environmental factors. So I think that from a perspective of genetic and environmental risk factors, the idea is that they may explain part of the missing inherited susceptibility. And genetic and environmental interactions mean that in someone who has a specific genetic makeup, the exposure to a specific environmental risk factor can increase the susceptibility compared to individuals who don't have this genetic makeup and they are exposed to the same environmental risk. So we can look at additive interactions, but also multiplicative. So if I take the example of additive interactions, it means that overall the risk for both factors combined is higher than the sum of the two risks if you take them separately. The interest is that the presence of interactions not only shed light on the disease processes at play in a given disease such as MS, but they can also result in giving options of preventive strategies. I'll take just one example. We have looked to pediatric MS, whether pollution with ozone in the air increases the risk of MS. And it increases the risk of MS, but individuals who are HLA-DRD1 positive, which is the strongest genetic risk factor, if we find that there is an interaction, which we did, then you could think, okay, I could maybe decrease exposures in individuals who have that genetic risk factors, so it would lower incidence of MS down the line. I think we're not able to do that yet because we're really at the beginning of figuring out genetic and environmental interactions. But that's something that in some cases could yield interesting opportunities.
I mean, I think it's clear already from the discussion so far that this is a really complicated area and trying to not just identify the different factors that are playing a role, but obviously how they interact and trying to narrow these down, as you say, retrospectively. I think one of the ways, and you sort of touched on it at the end there, one of the ways to try and get around this that you've done a lot is doing research in a cohort of people living with pediatric MS. Can you talk to us about, I guess, your choice in terms of doing that and how important you think a pediatric MS cohort is in terms of being able to understand these factors better?
Yeah, so it's there's a lot of positive and a little bit of negative. So I'll go through that. So adult neurologist by training, but I started to take care of pediatric MS patients more than 15 years ago. And when we started looking at pediatric MS, initially with the support of the National MS Society to start clinics in the US, and then now further support to do research, I realized that it was really a unique opportunity to study environmental risk factors for the disease. Although it's more challenging to reach large cohorts of patients that you can study, because luckily pediatric MS onset is more rare than adult MS onset, the opportunity is that the children have a shorter time between the exposures they have and the disease onset. So we are likely much closer to the biological onset of their disease. So that's one strong plus. Another plus is that the parents are typically around the clinic visits, and if we're discussing research, they are very much motivated to participate to understand why the disease started so young. And they remember the exposures that occurred not that long ago. If you work with adult patients, typically, onset of the disease will be in their late 30s or early 40s. And if you ask them what happened during childhood, lots of information are lost because people don't remember, their parents may not be around. If you work with children also, there's much less irrelevant environmental exposures because they've not had time to have that many exposures. So you may have a cleaner picture. And I would say also in terms of infectious disease exposures, children are still in an age range during which they are acquiring the very common infectious exposures, It may be easier to tease out the role of these infections in the disease.
Following on from that, and now going a bit more into the environmental factors, which we've touched upon already. You mentioned in infectious exposures, EBV has obviously been one that's gained a lot of attention recently as being a really large environmental factor that could be involved in really heightening the susceptibility to MS. Can you give us a bit of an update on what we know on the EBV story, but also what other environmental factors are the ones that are really of interest at the moment?
Yeah, so the situation with EBV is a very good example that highlights the fact that unless there are gene environmental or environmental interactions, one cannot explain why most of the world's population, which has been in contact with EBV by age 25, basically, does not develop MS. And a prior EBV infection increases substantially the risk to develop MS, both in children, but it also in adults. But it is not sufficient to lead to disease onset. So our group and the Pediatric MS Network in the US, but other researchers in Europe and Australia have started to look at the possibility of gene-environmental interactions to explain why EBV may actually be more strongly associated with MS or not. So one thing I can say is that in one of our earlier study, we found that there was a synergistic effect of EBV infection in those carrying the DRB1 genetic variants that I mentioned earlier, increasing the pediatric MS risk. So I'll give you some examples. So if an individual and this is from pediatric MS studies, so if one is DRB1 positive and they have been in contact previously with EBV, their risk to develop MS is multiplied by 15 or so. If they are DRB1 negative, then their risk to develop MS once they have encountered EBV is only in the ballpark of 3 or 4. So that you can tease out a little better in children, because when we see those patients, some of them are not positive yet, whereas when we work with adults, the vast majority are already positive for EBV. So there are probably other genes that are involved in the increased likelihood of developing MS related to EBV exposure. One of the genes, genetic variants that we have studied and others in Australia had studied is a gene variant that is on CD86, which is expressed on antigen-presenting cells. And there is also an interaction between that specific variant on CD86 and having had EBV. So the effect is basically, increased by 3 points or so when you look at that gene. And so it's interesting. It gives you an idea that antigen-presenting cells may be more important for the trigger of MS in those individuals carrying the gene.
I guess this is going to be a difficult question, a bit out of the off-center, but being able to look at pediatric patients and adult onset patients and thinking about an environmental factor like EBV, do we have any understanding yet as to, EBV exposure, for example, tends to happen during adolescence. There's obviously quite a lag phase from that in adult onset patients between that environmental exposure and development of the disease. Do we have any idea yet why, obviously in some instances we get this pediatric where, there's a less of a lag, and obviously EBV not the only environmental factor, but do we have any insights yet in terms of what may cause more rapid onset in that pediatric stage versus later stage?
So this is a multi-million-dollar question, and that's why people have chosen to study pediatric onset MS, because those patients have their onset 20 to 30 years before the typical age of onset in adults. So I believe, and other people believe, that it's possible that those who develop a very early onset in childhood may have either a higher load of exposure in terms of number of exposures or the magnitude of the exposure itself, and or a higher genetic load also. And then the combination of both may result in an earlier onset. But this is not proven. These are theories, so I want to be clear about that.
This has been the big thing since the EBV story, that there's still many questions that we need to answer in that space to really get an understanding of how exactly mechanistically it may be involved. I think one of the first things that anyone who I certainly remember 20 odd years ago when I started in the MS space, one of the first things we see is the gradient across the world and how we know that we have a lower incidence at the equator versus, the further away you move from the equator, more people, or the higher the risk of developing MS. Just wondering if we can touch on, is there any greater understanding around the environmental factors that may be related to that? Obviously, UV exposure, vitamin D have been the ones that have been hypothesized. Any sort of updates on where we are with our thinking of that?
So I think that the gradient historically was very clear. I think in more recent studies, the gradient is not as strong for a reason I think that are unclear. The reasons, as you touched on, could be relating to what kind of infections circulate at what age, what are the socioeconomic makeup also of a given population, making some infections earlier rather than later, or vice versa, and then the genetic makeup of a given population. So I think this is really hard to know exactly what's explaining the gradient that has been seen in MS.
As moving from environment onto genetics, as we sort of said in the intro, we've known about a genetic component of MS for a really long time, but we're continuing to learn more and more. As you say, we've got more than 200 variants now that contribute some level of increased risk to MS susceptibility. What's our sort of latest understanding of how genetics are contributing? What are the newest things that we've found in that space?
So larger studies allow to detect smaller genetic risk factors, and also studies of populations that are racially different also highlight what are the variants in a given population. The vast majority of the genetic studies have been done in whites because first, MS is most prevalent in white people, but also it's really hard to have a sample size big enough if you have a heterogeneous population to really address the genetic risk factors in different racial groups. And so people have selected a category of individuals that belong to a race where MS was more prevalent. So I think now there's more effort to understand what are the genetic risk factors in African Americans or in Hispanic patients, for example, or Asians. And I think that probably will unravel more information about the disease processes that are underlying MS.
And does it make sense to suggest that we're going to have slightly different genetic risk factors in different populations because they're also going to have slightly different environmental exposures, and so the genes that interact are potentially going to shift?
So there are genes that have different variants in different populations, but serve the same purpose. And so it's just a different flavor of the gene, depending on whether people are, Asian or they are whites or they're African American. So that's one thing. To my knowledge, I'm not aware of genes that were genetic variants that were detected in a population that were radically different. I think what is found is mostly a different variant of within the same gene, but it's not impossible that there could be genetic variants that are located on different genes, although probably not that likely.
So I guess we've had a lot of focus, and obviously the focus of the episode is talking about risk factors and susceptibility, but just shifting that slightly, there's been a number of studies recently that have shown a lot of these environmental and less so genetic, but we had our first genetic marker linked to progression found last year. But a number of these environmental factors, as well as increasing risk, also seem to worsen progression, smoking being a really good example of that. What are your thoughts on that, just as a general concept, the fact that we've got this increased risk, but also it worsens susceptibility, and what's our understanding of that at the moment?
I think this is really the next frontier to understand how all these genes and environmental factors may contribute to disease worsening. Although there's several studies that have suggested that cigarette smoking, obesity, vitamin D status, and comorbidities, for example, are associated with faster progression of disability or increased relapse rate, we have to remember that these studies of association, so they do not prove causality. And it's always tempting to think that we have found the culprit, but at the same time, it can be much more complicated. I'll take the example of vitamin D, for example. Among the risk factors I just mentioned, vitamin D is one that could be addressed with clinical trials of supplementation. And in well-designed clinical trials of vitamin D supplementation at the right dose, the studies failed at showing a decrease in the relapses or immune lesions on the MRI. So what it means that the studies that have looked at vitamin D levels in the blood may have actually found an indirect marker of sun exposure, for example, and your skin color. And so the proving causality for these environmental risk factors is really very tricky. So some are not amenable to translation into intervention in clinical trials to try to prove causality. I'll take smoking or obesity as examples because you cannot randomize people to obesity or to cigarette smoking. So it makes it really very hard. I would say that because some of these exposures are known to have a deleterious impact on health in general, not only in MS, I think it makes sense to try to address them in the clinic when we see our patients, telling them, you're fairly overweight. We think that overweight leads to smoldering inflammation that in turn may impact negatively the course of MS. In addition to increasing the risk of comorbidities like high blood pressure, cardiac disease that also have been associated with a worse prognosis of MS. So trying to address that individually with patients makes sense. Similarly, there's been associations between diet and the risk of MS relapse, which is, patients always ask about what diet should I be on to slow down progression of my disease. And I think that there are several studies that have been published that suggest associations, but so far we've not been able to confirm causality. But most of the diets that have been found to possibly have a good impact on MS are actually helping diets that are good for health in general.
So I guess in closing, and I already know this is going to be a very difficult question to answer, what are your thoughts on a potential in the future as we continue to get a better understanding of these environmental factors and genetic factors for them to be used, I guess, prognostically to predict the risk of a person developing MS? Do you see a future where, you may see someone before an onset of MS, but you're able to, suggest to them, look, the likelihood is X%, and also using this information to shift towards more personalized treatment approaches, which I guess you've just touched on there as well.
Yeah, the study of gene-environment interaction is our next frontier, so we can approach the individual risk to develop the disease. One thing that is difficult to keep track of is, if I take the example of sun exposure, is that, if you're 20 and you wonder about your risk of MS based on your genes, that clearly can be genotyped. But based on your sun exposure level, it's very hard to have one marker of how much sun exposure you've had during your teenage, for example. So some of these environmental risk factors are very difficult to quantify. And not only in terms of the intensity of the exposure, but the length of the exposure and at what time they were really important. But I still think that at some point we'll be able to approach a more personalized prognosis in terms of risk to develop the disease. In terms of personalized approach to treatment based on genetic and environmental exposure, once a diagnosis has been made, I think it's going to be even more difficult to address because clearly that part of the field is lagging and there's the issue of being able to determine causality or not of some of these risk factors. Either way, what it's going to do, it's going to take very large collaborations to advance the field of gene-environment interactions because we know we'll need to enroll many thousands of patients in these kind of studies.
Well, it's very interesting to think about where we may get to with all of this research, but I really want to thank you, Emmanuelle, for joining us today and for sharing all of those fascinating insights.
Thank you for inviting me.
And thanks everyone for listening. Once again, I'm Brett Drummond, host of this podcast episode. I'm also co-founder of MSTranslate, an independent resource centre that aims to simplify the complex wealth of information about MS for the greater MS community. The ECTRIMS podcast is produced and hosted in collaboration with two key partners, MSTranslate and the MS Journal, a peer-reviewed international journal that focuses on MS, neuromyelitis optica, and other related autoimmune diseases of the central nervous system. The ECTRIMS podcast is supported by an educational grant from Novartis, Roche, and Alexion AstraZeneca Rare Disease. Educational grant providers have no input on the podcast series content. You can read more about the topic of this latest podcast episode and more on the ECTRIMS website, ectrims.eu. Do subscribe to the ECTRIMS podcast on your favourite podcast channels and follow ECTRIMS on X, formerly Twitter, LinkedIn, Facebook, and YouTube.